Hi everyone, and welcome to today's lecture, which is going to be about the sensory contributions to motor control. And if at the end of this lecture you find that this is absolutely fantastic stuff, I really urge you to take neuromotor control processes. At the undergraduate level, that is kinesiology 636, and at the graduate level, it's 736, where, if you take it with me, we're going to go into a really deep dive into the neurons, the circuitry, all of that kind of good stuff in the peripheral nervous system as well as the central nervous system, more than you ever thought that you would want to go. So before we actually get started with the sensory input and what we'll be talking about, it's important that I define a couple of things for you first. The first is the concept of an open loop system. An open loop control looks very much like this, where you have an executive in the motor system that actually initiates a plan, and after that movement occurs. So in this case here, the central nervous system, or for example, which could be in the brain or in the level of the spinal cord, it provides output which is independent of any sensory input, so nothing is coming in here, or from any movement outcomes. Okay, so it's basically like throwing a jab. It doesn't rely on any sensory information that's coming in, but the executive must have a knowledge of the status of the system and its surrounding in order to make that movement plan. That's not to say that feedback isn't available to the system at this case. It's just that it's not going to be utilized during the activation of their movement program, which may be essential for the selection of the appropriate activity. When we start talking about speed accuracy trade-off, what you'll realize is that for open loop control, like we see here, oftentimes these are movements that occur in such a fast time period that you can't actually use any feedback, whether that be from the visual or the proprioceptive system, to actually control or to correct any movements. In contrast, closed loop systems can be of these two fashions. The first one, the level 2 closed loop control, is when the executive motor system actually makes a movement plan and receives feedback via the muscles, which it can use to either ensure the movement is progressing as it should or to correct a movement. And an example of this level 2 closed loop control would be, for example, a gymnast walking on a beam. A level 3 closed loop control is very much similar, except that you're going to get feedback via the brain, and this could also be from the visual system. And an example of this would be taking a shot in racquetball. So regardless of whether it is this closed loop control, you get feedback via the muscles or via the central nervous system itself, it's really important that the sensory input or the feedback is processed and it's used to generate or refine movement commands. And in this case, we can say that the central nervous system changes the output, so this part of the loop going this way, based on sensory input that it's receiving. And of course, in this case here, we have to understand that there's got to be enough time for this loop to be completed if we're to use the feedback coming from our sensory input systems. So, what we do know is for human motor control, as well as animal motor control for that matter, sensory information from both internal and external environments is essential for both the initiation as well as the regulation of movement. And when we actually are going to use that sensory information, we need to know three things. First is the state of the environment, right? So in this picture here in A, this would be like we need to have a model of the world, right? So we need to understand what is going on in the world. The second is the state of our body. So for example, down here in the comparator, all the way through here in the action and sensation, we need to understand what our neural and our mechanical system is looking like and how those actually occur. So in this way, for example, think about the neural state of the body being, as we are as healthy young adults, how that differs, for example, for somebody who has a stroke, right? So their neural structures are actually changed due to that disorder. Or vice versa, or conversely, it could actually be if somebody is old. And so when you start to get old, you get these deficits that occur both within the central nervous system itself, like in the cortical structures, you start to lose all of those things. But it could also be that you lose the muscle spindles, the sensitivity, and the sheer number of them. So we need to have an idea of the state of the body. And the third part is the state of the body with respect to the environment, right? So we need to know where we are, and we need to know where the environment are, and how those actually interplay with one another, okay? And so this is when we see these models. So we've got a very kind of schematic flowchart model here of how it would work. This model part B is more of this concept of the human body and putting the brain in the middle, okay? And the world is on the outside. And this one in here is actually kind of how I would demonstrate it in neuromotor control processes, where we're really looking at what are the neural processes occurring in the central nervous system at the cortical and subcortical levels that are controlling how that sensory information is received and then subsequently shunted over for our motor commands to take place. A good way to think about this is to use these models. And so purposeful human movement is actually governed by interrelated loops, both with respect to feed-forward loops as well as feedback loops, okay? And the brain employs these implicit models that are going to be trained through experience. So let's take a look at what we're dealing with here. So basically we have a multimodal state estimate. And basically this multimodal state estimate relies on vision, proprioceptive, and vestibular information to have a sense of our belief about the state of the body and the world. And as we're actually making our commands, we have our feedback control policy. For example, are we going to rely on vision or vestibular or both of those senses? And then we can actually make a motor command coming from the central nervous system, which is actually going to be placed into the body and the environment, for example, making a reaching movement, which eventually will lead to a change in state because your hand is actually moved from its initial position. But when we make that command, as it goes to the body and the environment, what the brain does is it makes an actual efference copy or a photocopy of what it's done, right? And by making this kind of facsimile copy of its motor command, it can actually use that to determine whether the predicted sensory consequences of what it's doing actually line up with, down here, the measured sensory consequences. And we use this information both from the predicted and the measured, and we actually get this difference between them. And we use that to feed back into this multimodal state estimate back forward again. So for example, so that we can actually change the movement if our predicted sensory consequences and the measured sensory consequences aren't the same. So because this is a lecture about the sensory contributions to motor control, we need to actually understand where the primary sensory inputs are going to be. So this is kind of just a slight change from the figure that I showed you on the previous slide. And so what we're seeing here is that multimodal state estimate, as I've told you, actually comes from primarily the visual, the proprioceptive, and the vestibular systems. These feed both into the multimodal state estimate, but also have a direct influence on the feedback control policy that we're going to use, right? And this differs depending, of course, upon our state of the world. So for example, if you've got pretty poor proprioceptive control, due to, for example, an injury or something of this nature, maybe fatigue, then you're less likely to use proprioceptive information for your multimodal state estimate, although you may use it for feedback control. So once we've actually understood where we are in relation to the environment and we start to make our movement, here's our feedback control policy, and we're going to make that motor command, right? Of course, it goes into the body and our environment, for example, making a reaching movement. And as we make that state change, right, so our position, which might be in our pocket and then ends up reaching for a beer, we can actually see this coming through in our three sensory systems. And after a time delay, because of course information has to be transmitted from the central nervous system down to the musculature, from the musculature up through the sensory systems, we get this sensory feedback that we can actually pick up in our three systems. And that information, again, gets actually compared to our feed-forward prediction model, our efference copy, which we can actually store either individually in our three systems or as a gestalt, which is a combination of all three. And what we do is we compare these state estimates from our efference copy as well as from our sensory feedback. And we use this information and compare it, and then we can actually change our state estimates here from our intramodal sources. And so that's basically, in short, in a very cursory view, how we use sensory information to first make our movements, but second, to actually adjust them or to determine whether we've made the right kind of movement. And again, I'm not going to go into too much detail about this inner circuitry because I cover it in great depth in 636, where I actually spend weeks and weeks just on this process alone. So we can actually conceptualize what we're talking about in today's lecture in this way. So basically, if we consider it this way, we've got our brain and our spinal cord, which is a part of our central nervous system, right? And this is where most of our motor commands are made. But what we're going to do is our body, if we consider it to be how we balance ourselves with respect to, for example, standing on the Muni, we get information from the visual system, from the proprioceptive system in our muscle, in our joints, and in our tendons, as well as from the vestibular system. This information actually gets combined into a multimodal system and comes into the brain, where it's actually, we make sense of it. And then after that, we can actually keep our balance. Unfortunately, damage to any part of this circuit, for example, in the visual system, in the proprioceptive system, in the vestibular system, or in the central nervous system, when that happens, what happens is you can lead to dizziness, vertigo, imbalance, or incoordination, okay? And so what we're going to do today is we're going to take a look at these three systems and how they might actually contribute to motor control. So we're going to start with vision, because vision is actually our preferred source of sensory information. The human visual system is actually different from the other systems in that it provides us with an extraordinary amount and also quality of information about the world. And we can actually achieve this by just a single glance, which may take just a few milliseconds. So when we look at an object, whether it be a biological object or a non-biological object, that simple glance can tell us information about its location, its size, its shape, its color, its texture, and whether it's moving. And when we think about vision in the control of our movements, it's actually essential through all three phases. It provides pre-movement information, right? So when I'm reaching for a bear, I need to know where the bear is if I'm going to reach it. Second, it provides feedback about the movement in progress. So as I'm reaching for that bear, it tells me whether I'm on target to reach the bear or whether I'm going to knock something over. And it also provides us with post-movement information about goal achievement. Have I reached the bear or not? And there's different ways that we can actually test the role of vision in motor control. One way to do this is by actually examining our eye movements, because our eye movements reveal a great deal about the strategies used to inspect a scene. And indeed, vision is this first idea of attention to something. So our eyes precede our hand movements, right? Because we have to understand where the object is before we can actually reach for it. Vision is not a passive process. It's an active process in which eye movements shift in that scene several times each second, and it shifts to parts of the scene that we find to be especially interesting. Okay? And as we're scanning that visual field, we pause to focus attention on the portions of the scene that convey the most significant information relevant to the task at hand. So if we look at this bust of Nefertiti, who was an Egyptian ruler, when we use eye tracking on it, and what we're doing here is we're looking at where the eye stops and the movement path that the eye is making, what you can see is that when people do this, those eye movements are actually focused on portions of the scene that are most significant. In this case here, we're looking at the facial structure, the eyes, the nose, and the mouth and chin area, as well as the ear, right? So we're not really paying attention. Nobody's paying attention down here to the stand or to the mid-neck part of it, right? And again, this depends on what you're looking at and what is appropriate for the scene in general. One of the primary questions in early motor control research, and when I say early, I'm going to take you back all the way to 1899, and R.S. Woodworth, when he was doing his PhD studies at Columbia University in the Department of Psychology, he was interested not just in the way that we think. He was interested in how does the way we think and the processes in our brain, how does that affect our ability to make movements? And then for his thesis, he asked specifically, how much time does a human need to process visual feedback in order to perform a closed loop movement? And what he did was he used a climograph or a memory drum that looked like this. And so what this had is here, this is a piece of paper, and the paper fits onto this reel here. And what you can do with this reel is it's connected to a motor, and you can set the motor at different speeds. And so this paper actually starts to rotate around here very much like toilet paper does, okay? And then he put this shield over it here, and so there was only this slit available, okay? And what he asked people to do was to draw lines on this climograph through this slit here of a required length. Okay, so for example, sometimes they had to make lengths of 20 centimeters. And because the climograph was actually rotating, right, as they're making these movements, it wasn't like they're making movements on a line of paper like this, where they could actually see what they were making before and correct their errors. As they were making the movement on the climograph, they had no information about what the preceding movement looked like. And so in his dissertation work, Woodworth actually asked people to do this with the left hand and with the right hand, and also with the eyes open and with the eyes closed. For the purposes of today's talk, we're just going to focus on the eyes open and the eyes closed condition, and what we're going to look at is how much error they had relative to the length that they were asked to draw. And so what I'm doing is I'm plotting here for you on the vertical axis, the average error in milliseconds. So in this case, it doesn't matter whether they overshot the target of, let's say, 20 milliseconds, or whether they undershot it. It's an absolute value, right? So in this case, they were 4 millimeters off target, but it doesn't matter the direction, okay? And on the horizontal axis, we have movement time. How long they were provided with information. So what you can see here is that when their eyes were closed, there wasn't really much of a difference in their average error, regardless of how fast the climograph was moving, okay? But what you can see is when the eyes are open, when you give people more time to process that visual feedback, they actually improve, okay? And what Woodworth actually did by kind of plotting it in this way, he found that about the first statistical difference you get is around here, which is about 250 milliseconds. And this is the point in time, and this answered Woodworth's question. He said, based on my experiment, the human system needs 250 milliseconds of visual feedback, 250 milliseconds in order to use visual feedback to control its movements. Now, 250 milliseconds is actually quite a lot of time. You think about that in sporting contexts, oftentimes in baseball and soccer, etc. You just don't have that pure time. And so a lot of people thought to themselves, well, this seems to be a overestimation of time, this 250 milliseconds. And so what my advisor, when I did my PhD, Howie Zelaznik, what he did when he was a postdoctoral student at the University of Southern California, he actually said, I think that's an overestimation. And one of the problems with what Woodworth did is that you either had your eyes open and your eyes closed, but you always knew that you were going to have that information. And so he said, vision, when its presence can be expected, can be used in far less time than the minimum suggested by Woodworth. And what he did with his future wife, Lorraine Kisselberg here, who was a student at USC at the time that Howie was there, they did this wonderful study where when people were making these movements, they suddenly switched the lights on or they switched the lights off, right? And so they actually played around with the uncertainty about whether visual feedback is available on that given trial. And so by doing this, you can see that when the lights are off, there doesn't seem to be any difference in the amount of error. Okay, it's around 3 to 5 milliseconds. 3 to 5 millimeters. But when the lights can actually be turned on, you can see that you get this improvement that the error rates actually go down. So you get better with more time that the light is on. And so based on this information, Howie said that you could actually use visual information to guide your movements in as short a time as 100 milliseconds at this point here. Now, one caveat that we should know about this is that, of course, it depends on what movements you're doing and how much light availability you have. And I know that we talked about in our previous lecture about human information processing, about the clarity of the stimulus as well as the intensity of the stimulus, right? So, of course, the more clarity that the light affords you and the more intense that the light source is, the more likely and the quicker you'll be able to initiate that feedback loop. Versus, for example, if you just have a little bit of light available to you, for example, in candlelight conditions. And what was really interesting is this different way to look at this problem. So how we looked at it by actually switching the lights on and off, right? So playing around with uncertainty. Another way that you could do it, and this was done by Whiting and colleagues in 1970, where they looked at how much time is required to use visual information for intercepting movement targets, okay? Whereas Howie looked at just to actually make a discrete movement. So this would be akin to, grabbing a baseball or something that's thrown at you. And here's how the paradigm worked. Here's the apparatus. They had a ball that was stuck up here at the release point. And then the ball would be dropped from a 3-meter height onto this angled trampoline-like spring, and which actually bounced the ball another 3 meters towards the subject who attempted to catch it. And the trials were actually conducted in a completely darkened room. But there was a small bulb inside the ball that could be lit for periods of 100, 150, 200, 250, and 300 milliseconds after it hit the trampoline. Okay? And so over here, if we look at the results, when you have full light, right, so in a regular room with the lights on, people catch the ball about 95% of the time. So that's kind of like the baseline level of performance. And what you can see is that as you get more time, more light available to you, your actual performance increases, okay? So on the horizontal axis, it's the time before attempted catch without vision. So this is 300 milliseconds without vision. So you're about 30% levels, which is worse than chance. If you only have 200 milliseconds, you're about chance levels, so 50%. And when you have only 100 milliseconds without vision, which is akin to having 300 milliseconds with vision, you can see that you're up at 80%, which although it's not as good as the full room light condition, it's still pretty good. So, although never as good as in full light, visual information of the ball flight could be used in at least 100 milliseconds to improve catching performance.